Hi, I am Mrs. Sloan, and this video is intended for AP biology students. And today we are going to be talking about viruses, bacteria, and archaea. So I'm going to make myself a little bit smaller and present. Remember the notes, if you would like to have them like my students have them, they are located down in the descriptor of this video, 2 columns. Column 1, I'll help you fill those in. That's the scaffolding of the information we're going to talk about today. Column 2, I encourage you to throw in pictures that are helpful for you. All right, and there's the coronavirus right there. We're going to get to that. Okay, so for my students, this is chapter 20. This right here is the AP expectations for Unit 6, Gene Expression and Regulation. And so the topics that we're going to be discussing today come from 6.4 translation and 6.7 mutations, and also we'll do a little bit of review of bacteria as well. All right, so let's get started. So viruses, pretty small. They are known as obligate intracellular parasites or a microscopic pirate. They are associated with disease and they are very host specific. Now some viruses can species jump from one, let's say a bat, to a human, potentially. That would be a species jump. But some, like tobacco mosaic virus, it can only infect plants, in fact, only tobacco plants. So on your notes, what is a virus? It is associated with disease and it is host specific. That's the first part you need to fill in there on your notes. They are not considered living, and here you can compare and contrast viruses with prokaryotes. So viruses are not made out of a cell, right? And living things are all composed of cells, and viruses are not made out of cells. They have no metabolism on their own. They don't respond to stimuli. They will only multiply inside of a host cell. But because they have a nucleic acid, they can evolve and evolve very quickly because they can mutate. So you want to put on your notes right there. I think the part you need to fill in is right up here at the top, that they're obligate intracellular parasites, have a nucleic acid and can evolve, but require a host to replicate. They require a host to replicate. And if we were looking at the size, sorry, having trouble moving. I'm going to be bacteria. If we were looking at the size of them, they are smaller than a ribosome. Remember, a ribosome is a workbench for protein synthesis. And there's 2 components that they have. They have a protein shell for sure, and they have a nucleic acid. They may have some other things, but for sure, all viruses have a protein shell and a nucleic acid. Now, when you look at their nucleic acids, they have a tremendous variety. I mean, it's either going to be DNA or RNA, but what I want you to look at right here is double-stranded DNA, like what you have, right? And you can have a DNA virus, but you can also have an RNA virus, and that RNA virus can be single-stranded or it can be double-stranded. Now, remember when we do replication and transcription where you use DNA to make RNA, right? Central dogma. The RNA that we make in our cells, right? It's going to read from 5 to 3. That would be what you would call a plus strand right here, meaning if you're an RNA virus that is in the right direction, that it can be read by your cellular components to build a protein off of it, that would be a plus. A negative strand virus would have to be replicated into a positive strand, mRNA, that could be translated. Now, here you have a double-stranded virus. Here is a retrovirus. This is doing the central dogma in reverse. So what they have the ability to do is they have an enzyme called reverse transcriptase, where they can make a DNA copy of their plus strand RNA virus. All right? And they can also be single-stranded DNA viruses. So you have a whole host of whatever you can imagine for nucleic acid. That is what they have. So where did these viruses come from? Well, they could have evolved before the 3 domains. Bacteria, archaea, and eukarya. Eukarya are the 3 domains. Remember, bacteria and archaea are both prokaryotic cells, eukarya, eukaryotic cells. or since they're so host specific, maybe they evolved from their host. And their classification, remember the classification, domain, kingdom, phylum, class, order, family, genus, species, they start at order level of classification into their different groups. Now, how are they classified? Well, I'm going to come back to that picture right there, but usually by the type of nucleic acid they have, and then I'll go back to that picture, what size or shape they have, and go back, sorry, I'm moving around too much. What size or shape they have, and whether or not they have an outer envelope. Okay, so remember, they always contain a nucleic acid. So here you can see this classification. I am not moving myself. There we go. Sorry. This classification, thank you. This classification shows you whether it's a DNA virus or an RNA virus. Notice how they've broken it up. A double-stranded DNA virus like herpes, and it has an envelope, right? Or a single-stranded DNA virus. Here are all your RNA viruses, whether they're positive, minus, or both. Okay, and you can see the different ones that are listed here, but the one you might think about right here is the coronavirus, right? So it's an RNA virus, and it's a plus, so it's one that can be immediately translated in our cells, okay, immediately translated, and it has an envelope on it, and there's several different coronaviruses. The one that we are dealing with right now is called the novel coronavirus, and we'll talk about why that is in just a little bit. Okay, so now you can see the virus particle, the inner core, will definitely have nucleic acid, DNA or RNA. It could have various proteins like a retrovirus is packaged with reverse transcriptase, an enzyme that makes RNA go back to DNA. And then the outside will have that protein capsid, and it may or may not have this outer envelope on the outside. And those are the ones that could have the outer envelope could have the spikes on it. And those spike projections, the reason why the coronavirus is called that, because corona means crown, and these little spikes on the coronavirus kind of look like little crowns. So classification by size and shape, on your notes there, type of nucleic acid, and if there is an outer envelope, they have virally encoded protein spikes that allow them to gain entry to a cell, the cell. Their outer structure, protein capsid, nucleic acid inner core, and then I gave you the envelope or not. Let's take a look at a few. This is the adenovirus. You can see right here, it's got the outer capsid. It's got, it's a DNA virus on the inside. We've talked about a bacteriophage before. You can see the capsid and the nucleic acid inside. Remember, bacteriophages infect bacteria. Here's that tobacco mosaic virus. It's an RNA virus with its protein capsid. And this is the influenza virus. This is an RNA virus, and its protein capsid is inside. Here's its envelopes, and then you can see these spikes. And this is typical when you're getting into through a cell membrane, is those spikes. There is some receptor that they're taking advantage of on our cells that then allows them to gain entry into our own body cells. All right, so how do you study viruses? In some cases, make vaccines. Just so you know, the vaccine for the coronavirus is not made through eggs, right? It's an mRNA, and its code, it's encoded in, enclosed within like a fatty layer to get it into our cells. And what it codes for, that mRNA, is for those viral spikes. So we'll start making that so it will trigger our immune system. But vaccines can also be made within eggs because that's a great host, this egg, to grow up those viral particles. All right. And the coronavirus. So let's talk a little bit about that. I will go over here. Okay. So It is an RNA virus. You can see that, but it's also associated with some proteins with that RNA. It is its capsid that has its spikes on the outside. You can see the spike glycoprotein. That's why it's called corona. It can cause diseases. There are several, and I gave those to you in your notes. There are several different diseases that can be caused by it, and it can species jump. This is called the novel coronavirus, or COVID-19. And the CO stands for corona, the VI for virus, and D for disease. And then in 2019 is when this was first identified. Now, there are so many different coronaviruses, and these are ones that have caused different infections. And you can see the different years here that they have caused those infections. And then also you can see the species of origin, like its natural host. So you can see rodents and bats come up several times. Here, SARS, the coronavirus that we're looking at right now, this right here, it's the ACE receptor that coronavirus spike is using to gain access into our cells. So a couple of things about this coronavirus is we're worried about these spikes, or the concern about these spikes is because this is the part in some of the newer strains, at least as of this time, this is January 2021 that I'm talking. These are rapidly mutating the proteins of those spikes, and then some of them, it makes it more transmissible and those spikes are making it easier to get into our body cells. So here are the different proteins that are associated with the coronavirus. So you can kind of see their different functions. So the spike protein, that particular protein, is critical for binding to host cell receptors to facilitate entry. And then there's some other descriptors here. And then you can see, and I need to get a pointer, and you can see the spike proteins right here. All right. And I gave you this in your notes, but reminding you, I gave you the part about the coronavirus, but I want to jump ahead in our notes. If you go to reproduction of viruses where it says APBio Mister, that's just a little mnemonic to learn how viruses can their viral life cycle. And we're looking at a bacteriophage right now, if you can recognize that. So the first step is to attach to whatever cell it's going to infect and take over. Penetration is however it gets its nucleic acid into that organism's cells. Biosynthesis, remember, you're going to have a leading strand or a plus strand mRNA that can be translated to build the viral components, whether it is the protein capsid or any enzymes that it uses. So that would be the biosynthesis stage. Maturation stage is building when your own cells, now when they're done building the proteins that the virus needs, that it starts to put the viruses together and then release. Now here it's lysine of bacterial cells, but there are other ways to kind of bud off of animal cells. So there's different ways, but that virus then gets out and spreads. So the way my students remember it is APBio Mister, APBio, and then Mister, maturation and release. And let's put the notes in right now. So attachment is a host-specific match for your notes. Attachment, host-specific match. Penetration, it is either engulfed or it injects its nucleic acid. It is engulfed or it injects its nucleic acid. Biosynthesis, you hijack the host machinery to synthesize viral components. Hijack host machinery to synthesize viral components. Maturation, you assemble new viruses. And release is exit through lysis. In this case, that's what's going on, lysis or budding. And we'll look at some different modifications of that. So back to the coronavirus again. This is from the New York Times. This, if you click on this, if you download this slide presentation off the notes, there's a link to it. You can read that article from the New York Times. And this is inside the newest variant that people are concerned about right now. And here you can see the genome, and they've got it kind of color-coded right here. You can see the spike proteins are here in red. Okay, and there's about 30,000 letters. Now, you and I know what those are. Those are nucleotides, right? And to assemble 29 different kinds of proteins. So the mutations that they're concerned about is changing that spike protein. And these mutations come about randomly. They could be selected for based on drugs that we are giving people. So how did this variant evolve? It could have, when the virus sits in somebody that maybe is not showing critical symptoms, it has time to just evolve within that and replicate within it over several months and accumulate new mutations each time. And then depending on what drugs you're getting treated with, it could be a form of natural selection or directional selection so that they can escape, and the ones that are surviving and reproducing are that are continuing to put more of themselves are actually selected for by the drugs that we're putting into our body. I'm not saying not to take those drugs. I'm just saying over time, that happens also with antibiotics, right? That people who are taking a lot of antibiotics, or as a society, as we take a lot of antibiotics, that can be a selecting factor so that stronger strains of bacteria are surviving and reproducing. That is what life does, right? It tries to succeed. So this virus, it's not a living thing, but what environment it is exposed to, some virus strains will be knocked off and some won't, and those are going to be able to continue and infect other cells in our body. Okay, so in class, we're going to watch a quick video on the coronavirus. You're welcome to click on this link if you download this presentation.
In December 2019, the Chinese authorities notified the world that a virus was spreading.
Okay, so next what I want to do is look at variations on viral reproduction. You have that there in your notes. So basically, in a short summary, there are two kinds of life cycles, lytic and lysogenic. Okay, in a lytic cycle, what happens is, look right here, you can see the bacteriophage infecting, and you can see, so you've done attachment, penetration, biosynthesis, maturation, and release, right? Remember all of that? That's your standard lytic cycle. Or it can have another, it can attach and penetrate, but it can become part of the host chromosome. And when it's doing that, it's called a prophage, and it enters into a lysogenic cycle. And it can lay latent, and every time that cell reproduces, it's reproducing part of that. It's reproducing that viral genome as well. So on your notes, we have bacteriophages, two alternative life cycles. The lytic cycle is the standard 5 steps. We're seeing them right here. Let me go to another one. Here we go. Okay, so here are your standard 5 steps: attachment, penetration, biosynthesis, maturation, and release. Okay, and on the last part of that, the host cell is lysed at release by virally encoded enzymes called a lysozyme. A lysozyme in order to cause that release. So Lysozyme. And we make that for the viruses because they have the messenger RNA, right? This bacterium is making it, and it's providing all the amino acids that it needs to build that enzyme in order to lyse its own cell. Now, a lysogenic cycle, this is what follows penetration, and then you have a step called integration. So following penetration, integration step where viral DNA is incorporated into bacterial DNA, and then that virus is going to lay latent, and it is called a prophage. And I would highlight that. I've given it to you in your notes, but I would highlight prophage, which is replicated along with the host DNA, And then different environmental factors can stimulate it to leave the lysogenic cycle and re-enter into the lytic cycle and start biosynthesis. For instance, if you get cold sores on your lip, things like the sun, if you're out in the sun, it can cause the damage to to your lip cells right there, if you have that virus, then it will stimulate the damage to those cells. It's like they're looking for another host, so they come out of their lysogenic cycle and go into the lytic cycle, and that's why you would get a cold sore on your lips. So environmental factors like UV radiation can trigger a prophage to re-enter the lytic cycle. And then sometimes the prophage causes the bacterial cell to produce a toxin, which can be bad for us if this bacterium is infecting us, right? So let's look at some different types of viruses. Let's start with an animal virus, right? Remember, we don't have a cell wall, just cell membranes. So viruses tend to be like a wolf in sheep's clothing. They've got some of our own membranes encapsulating them and protecting them, and they're trying to like, I'm just a little body cell, don't attack me. And so here they're using their viral spikes in order to gain entry, and then they kind of get uncloaked, and then whatever their nucleic acid is, you know, it might be a positive or negative RNA, might be double-stranded DNA or single-stranded DNA, but eventually it's going to be translated, that nucleic acid, to form all the things that that virus needs. And then as it matures, then it's going to get packaged. And then in this case, it's just a budding off of our own cells. So on your animal virus, I gave you quite a bit of it, but I said in the second sentence underneath animal viruses A, then can be uncoated and the viral genome can enter into a lytic or lysogenic cycles. Lytic or lysogenic cycles, and then B, can bud from host using various membranes, cell, Golgi, or nuclear membrane, or just lyse the host if it's a naked virus, or just lyse the host if it is a naked virus. So a virus that we talked about quite a bit before the coronavirus was HIV, and that is a form of a retrovirus. So here you can see it's got its spikes, which allow it to gain entry. It is a single-stranded RNA virus. Notice it's a plus, so that means it can immediately get translated. It's in that form, and it uses reverse transcriptase. So you're going backwards on your central dogma, going from RNA to DNA. It becomes double-stranded. There's a different enzyme that it is packaged with. Besides reverse transcriptase, it's packaged with integrase, which helps it to insert into our own body cells and lay hidden as a provirus. Then when it's ready, it can be transcribed and translated, and you can make more of those viruses, and then they will bud off right from our own body cells. All right. So on retrovirus, you have animal virus with RNA genome that is converted into DNA within the host cell by an enzyme called reverse transcriptase, can remain latent for years. And then the antiviral drugs interfere with these types of enzymes right here, anything that it needs to promote to the next step. And it can interfere with its life cycle. Kind of scary, huh? So let's look at some different viral diseases in humans. You can see there's several sexually transmitted disease like HIV and genital warts and genital herpes, mumps and measles, chickenpox. Those are all viruses. Look at the common cold, influenza, SARS, skin diseases. You can see here, digestive tract diseases, nervous system diseases like polio. And then here's some other wonderful diseases you can get from a virus. Did I say wash your hands? Wash your hands. All right. So an emerging disease. Now, this is not usually a novel one, but maybe conditions change or it's mutated or due to the type of travel that we have, it's now spreading. So causing a new or previously uncommon illness, and now it is a widespread disease, corona. So I said examples, HIV, West Nile, Hanta, Ebola, add on there, corona. And then some of the ways that it can move about, if you look on the cause, a virus can extend its range, gaining access to new area through transport or genetic mutation, or increasing their virulence factors. So some mutations allow a virus to species jump, like from a bat to a human. So some terms you want to know about is pathogenicity, and that is the ability of the organism to cause a disease, And then how badly, can it cause a disease, but how badly it does that is its virulence is the quantitative measure of its pathogenicity. And this is what it is based on. So there is a website right here if you wanted to go to, John Hopkins. And I just took a screenshot today of the global cases and the global deaths of COVID currently. But this is a link. So in the future, if you're watching this video, you can click on this link and it'll take you to the most current data on the coronavirus. All right. And so just want to reiterate, where are these things coming from? They're not usually new. So viruses have a really high mutation rate. And so with that, they can gain new abilities to get into living cells. There's a high degree of global travel, so people aren't isolated anymore. And so if it's in one location, it's easily spread to another location. There is no wall you can build high enough to keep a virus out. And then another one you need to consider is climate change. And this is a really interesting study. I will make this a link from Harvard about making connections between climate change and infectious diseases, some like malaria and dengue fever. And this is spread by mosquitoes. And so when temperatures change and it gets warmer, it's more likely for those mosquitoes to survive and thrive and then be a carrier of that infectious agent to us. Oh, in the notes, there is a link to it if you download the notes. So there is a link to it. Okay, then there's still other infectious agents. One of those is viroids. This is the smallest pathogenic agent, and this is just a naked piece of RNA. It does not have a protein capsid. It seems to only impact plants, and it directs the cell to produce more of itself. So on your notes, naked strands of RNA direct cell to produce more, and it can cause crop diseases. Now it is single-stranded, but it can fold and be self-complementary. So it looks like it's in a rod shape, but it's just like a circle that you smish down into a rod shape, and it can cause severe crop diseases. So this is without a viroid, and this is with a viroid. Okay, so direct cell to produce more. Then we have prions. So prions are misfolded proteins. And so that's why it says rogue shape. So here's, let's say, a normal shape of a protein, and here is a misfolded protein. And the problem is, it's a chain reaction. Once is misfolded, another one is misfolded, another one is misfolded. And so if you've heard of mad cow disease, and it causes basically the brain to become spongy and have holes in it, they're lethal for sure. Creutzfeldt-Jakob is one that humans might have. We wouldn't have the mad cow. And this category is called TSC, transmissible spongiform encephalopathies, and it basically causes holes in your brain. So on your notes, you want underneath prions, number 2, once infected, starts a chain reaction that converts other normal proteins to prions, and cause of TSC, and down at the bottom, like mad cow disease, All right, now I'm going to start talking now about a little bit about bacteria. I want to, this is mostly going to be review because we've already talked about bacteria. The virus part here was new. Just for perspective, bacteria have been around in microbes longer than anything else on Earth. So if you think of a 24-hour day, right, like this circle right here is 24 hours, microbes appeared at 5 a.m., dinosaurs appeared at 10 p.m. and the last few seconds is when humans appear. So on your notes, they were alone on Earth for about 2.5 billion years, so they became extremely diverse in their environments and in their metabolic capabilities. And as far as what our understanding of them, Leeuwenhoek, the father of microscopy, was the first to actually see them and to visibly see them, because people thought, because I can't see it, it doesn't exist. I'm sure you have heard of Pasteur. And Pasteur's experiments showed, people used to think that life could spontaneously come about. And so they would think that like rats came from garbage and flies came from rotten meat. And that if you just had a broth out there and you could see fungus and things growing on it, that was just this spontaneous generation of life. So here are two hypotheses and Pasteur in his experiments. So hypothesis A says bacteria arise spontaneously in broth. We know that's not true now. Hypothesis B say bacteria are in the air and contaminate the broth. So the first experiment that were done is that he would boil it, but he kept it capped, and then the flask outside, if you opened it briefly, then you would get some growth. Now, you would get no growth if you boiled it and didn't open the cap, but people said, well, the reason why is because you needed this life force from the air, not realizing that there was actually organisms in the air. And here, boiling to sterilize the broth, flasks inside a building open briefly showed less growth. So the conclusion is you would support hypothesis B. The second experiment, the swan neck flask, where he would boil it, no cap at all to interfere with the force of the air. And so, but what would happen is the bacteria would collect in the bottom in this dip and never get into the flask, and they would have no growth. So that supported that bacteria in the air is what contaminated that broth. And I gave you all the notes on that. All right, real quick here, let's review the structure. You have already learned this, but I just want to use this as an opportunity. So you know the cell envelope, you know about the glycocalyx, the carbohydrates on the outside, cell wall. This cell wall, you want to add to your notes, prevents the prokaryote from bursting or collapsing. Prevents prokaryotes from bursting or collapsing. You know, a cell membrane decides everything what goes in and out. The nucleoid region right here, that's the region where the DNA is located. There's not a nucleus, right? And on your notes, area where circular DNA chromosome is located. And then you also want to remember they could also have plasmids. You know, ribosomes are for protein synthesis. Thylakoids are membranes for photosynthesis. Flagella, that is for movement. You want to add that. And then a sex pilus, that is for conjugation. That's bacterial sex. They extend a sex pilus and exchange DNA across that, used to transfer DNA in the form of plasmids. And then fimbriae are for surface adhesion, Now, here's a picture so you can review all of those structures. This is a link right here. If you click anywhere on here, it's kind of cool. If you're interested in microbiology, there's a lot of information here, and you can look at different types of microbes. And here it's just showing the flagella, the plasmids, the fimbriae. You can see the flagella here and the fimbriae. Remember, this is just a review. We've already gone over this. When we talk about bacterial replication, it is by binary fission. And the only part you need to add in there on your notes underneath binary fission is, remember, it's clone offspring. And because it is haploid and it has a short generation time, mutations are generated and passed on quicker, and natural selection is quicker. So mutations are generated and passed on quicker, and natural selection is quicker. So how do you get variety besides mutations, right? So we want to look at some genetic recombination in bacteria, and there's three I want you to look at. So, and this is besides mutations, right? You can get conjugation, through a sex pilus, you can exchange. So conjugation, use pilus to pass a plasmid. Transformation is when you could pick up DNA from your environment and bring it in. The host absorbs the DNA from the environment and gets new traits. And remember when we looked at the S and R strains that were infecting mice in Griffith's experiment? Okay, that was an example of transformation. Artificial transformation is what goes on when you're genetically engineering bacteria. And then transduction is when a virus acts as a vector. So in the process of a virus infecting a host and becoming a prophage, that it could bring genes from host 1 into host 2. So transduction, virus acts as a vector of new DNA. Virus acts as a vector of new DNA. And transformation, the last word you needed there was environment. Bacterial metabolism. I'm sorry. I jumped over, characteristics on the cell wall. So there are gram-positive and gram-negative bacteria, and that's how well they take up the stain. So gram-positive, you can see this peptidoglycan layer of the cell wall is very thick. It's the outermost layer. It takes up the stain. That's a gram-positive. Gram-negative, the wall is in between two layers of membrane. It's much smaller, so it doesn't take up the stain. So all bacteria are either gram-positive or gram-negative. So what you need to know about gram-positive, the only part you need to fill in your notes there underneath cell wall is it is thick-walled and it traps stain and it stains purple. Gram-negative, they stain red, and just medically speaking, and I gave you this in their notes, this outer membrane makes them more resistant to antibiotics if it's a gram-negative strain bacteria. And so those type of infections from a gram-negative bacterium are typically harder to treat. All right, how are they classified? I put that in tiny letters because I don't think they're going to ask you about this on your AP exam, but different shapes right here, and then whether or not they cluster or not, but I gave you that in your notes already. Okay, and here you can see this is a gram-positive bacteria, and here is a gram-negative bacteria. They both have that glycocalyx on the outside. Okay, now how they relate to oxygen issues. So I gave you most of this, so I think I'm just going to review it with you. So obligate aerobes means it needs to have oxygen in order to survive. So you can see it's the only bacteria are surviving up here where there is air. Obligate anaerobes, they only survive if there is no oxygen. Facultative anaerobes, either or, doesn't really matter so much for them, and that would be an example would be E. coli. And then aerotolerant anaerobes, they don't use oxygen, so oxygen is not an issue ever. And I gave that to you there. All right, and then their metabolism, when you look at that, you can look at the different classification terms. So do they obtain carbon elsewhere? No. So they get their carbon and use energy from light to get their carbon. They are called photoautotrophs. If they don't get their energy from light, then they are chemoautotrophs. Maybe they're breaking down something like hydrogen sulfide. If they obtain their carbon elsewhere, yes, but they get their energy from light, they are called a photoheterotroph if they get their energy from light, but they are still heterotrophs. And then the other one would just be a chemoheterotroph. Now, on your notes, go underneath autotrophic bacteria, photoautotrophs, you have it, use light. Chemoautotrophs oxidize inorganic compounds, inorganic compounds to gain energy and to reduce their carbon dioxide, Now, heterotrophic bacteria obtain organic nutrients. Some could be saprotroph. It's a type of chemotroph that acts as a decomposer in an ecosystem. Now, you have heard of chemoheterotrophs because you are one, right? Animals are chemoheterotrophs. Saprotrophs, it's a type of chemoheterotrophs, and they will break down nutrients. Some are detritivores like worms that can break down fecal material, urine, and those types of things as well, right? Things that might be found in a cave and break down dead and dying things. So we'll talk more about those when we talk about other ecosystems. These are all prokaryotic products, vinegar, we've used soy sauce, right? These are all different types of prokaryotic products. Quick review. I'm not going to spend a lot of time here on symbiotic relationships. Just wanted to remind you of it because we do have symbiotic relationships with bacteria. So if you look here, that's just a model of the three different types. We'll talk about that in class. Cyanobacteria is, a a great bacterium, one because of the oxygen revolution, but also gets in these root nodules. And what they do is they do nitrogen fixation. Remember nitrogen fixation? It's getting nitrogen in a form that plants can use. And so these little root nodules are homes for that cyanobacteria. And what happens is the bacteria give the plant nitrogen in a form it can use, and the plants give the bacteria sugars that they can use. So that would be a form of mutualism. Commensalism is the bacteria that live, if you have a vagina, inside your vagina or your urethra. We both have urethras. They probably could be mutualistic. They don't do any harm to us, then it's commensalism, but they could also be mutualistic because bacteria that are not harming us are taking up space in our, let's say, urethra. And so that bacteria that are harmful don't have any access to it, don't have room to get into those areas because the good bacteria have already taken up all the real estate within those parts of our bodies. Okay, a parasitic bacteria is something like salmonella that you would get from food poisoning. And then here are just some various infections. If you're interested in these, you can just pause and see all the different types of diseases that we can get from bacteria. All right, now here's an endospore. The reason why this is important is because think of it as like bacteria in hibernation, and so they can still be, put in the right environment, like anthrax can go into an endospore, but put in the right environment can still cause harm in our bodies. Certain pathogens have been used as weapons, like anthrax and smallpox and the plague and botulism. I certainly do not promote that. A big concern about certain bacteria living in our body is that our prolific use of antibiotics is a form of artificial selection, which is then killing off the weakest bacteria, and stronger strains of bacteria are surviving and thriving, and we may not have antibiotics to protect ourselves against them. The purpose of antibiotics is either to interfere with a bacterium's ability to synthesize proteins or to repair or create cell wall. That is how they work. And I gave you all the notes on that. All right. Failure to address this problem of antibiotic resistance could result in 10 million deaths by the time 2050 rolls around and costing a tremendous amount of money. Now, a bacterium that you want to go yay is cyanobacteria. This was probably one of the bacteria that contributed to the oxygen revolution about 2.3 billion years ago, and that changed our planet. On your notes, cyanobacteria are gram-negative bacteria that are probably responsible for the oxygen revolution. They are also nitrogen-fixing bacteria, and there are many symbiotic relationships like lichens. Can cause an algal bloom, though, and that is not good. Here's the role in agriculture. Definitely for that nitrogen fixation, and you can kind of pause here if this is of interest to you. Here is just a classification of different types of bacteria. You can see them here. Here is cyanobacteria. And then here is Archaea, also prokaryotic, and it is our last topic in this chapter. And Archaea is considered prokaryotic, but it is different from bacteria. The cell walls are different and their cell membranes are different. Archaea are more similar to eukaryotes because they have similar rRNA to build the ribosomes. They too have introns and they have histone proteins associated with their DNA. What is unique to Archaea is they live in these really extreme environments. And I gave you those notes already. Here you can see universal ancestor, right, Luca? And you can see that from this universal ancestor, you have bacteria, but then you would put Archaea are more, though they are prokaryotic, they are more closely related to eukarya. And then you can see some differences on, like I told you before, the cell walls are different, but cell membranes are different. In that way, bacteria and eukarya have more similar cell membranes. And you can see here a phospholipid bilayer here, which you can recognize. And then this is the phospholipid bilayer that you would see in Archaea. Notice how it's continuous and it's not built out of fatty acids. All right, so some of the categories of bacteria, of Archaea, are methanogens, and they produce methane. So methanogens are chemoheterotrophs. They are obligate anaerobes. They produce methane, and maybe they will be found on Mars. We've got a Mars landing coming up, so maybe they will find some methanogens there. Halophiles are found where they're aerobic chemoheterotrophs. They can live where there's really extreme salt concentration, and they do a very unique form of photosynthesis. Thermoacidophiles are chemoautotrophic anaerobes living in very hot acidic environment like what you would find in Yellowstone. And then you have moderate environments, and they are in symbiotic relationships, but remember they are never parasitic, never parasitic. They do not cause disease, whereas viruses and bacteria do. Archaea do not. They do not cause disease, and they may be a major producer of nitrite in the oceans. All right, we did it. Good job. And I stay safe, stay healthy. Make sure that you are washing your hands. Make sure you're wearing gloves, right, when you need to, and you're keeping a mask on your face. I would like you to stay healthy. And if you're one of my students, I'll see you in class.